Hello everyone, I am Salim Mohammed, a master's student from Bowling Green State University, working under Dr. Angelica Vasquez Ortega and Dr. Kevin McLooney from the Biology Department. My research is titled Biological Cycling of Phosphate in Soils Amended with Dredge Materials from Toledo Harbor using enzyme gene expression and oxygen isotopes. To start with, agriculture is considered to be the backbone of the US economy because it contributes significantly to the country's GDP. In 2022, agriculture is said to have contributed 5.5% to the country's GDP and employment. According to the UN report in 2022, population growth has escalated the demand for food production in the past 50 years. Because of this, there is the need to pay keen attention to environmental sustainability and food security. This has led to a higher cost of fertilizer, reflecting an intensification of agricultural practices, posing challenges in the agricultural sector. One of the most important factors in the agricultural sector that has gained maximum research attention is nutrient cycling. It is important because it helps in determining the overall soil health. Now, phosphorus is a limiting nutrient needed by plants and animals for growth and development. Naturally, phosphorus occurs or exists in the soil as organic and inorganic forms. However, it is only the inorganic phosphate forms that are bioavailable for plants to utilize for growth and development. When phosphorus gets into the soil, they are being transformed into different types of pool. These transformations can be categorized into two main aspects. We have the chemical transformation and the biological transformation. To start with the chemical transformation, we have processes such as adsorption, precipitation. All these are chemical transformation that phosphorus goes through within the soil. However, for the focus of this research, we are capitalizing on biological transformation of inorganic phosphate within the soil. Now, with inorganic phosphate transformation, mineralization is a key aspect where microorganisms secrete enzymes such as phosphatases, which helps to hydrolyze organic P compounds to release inorganic phosphate compounds within the soil. The next one we can talk about is immobilization. With immobilization, microorganisms are able to incorporate organic P into their biomass. However, when they die, all these inorganic phosphate pools return back to the soil. Now, the role of microorganisms in inorganic P transformation in the soil is key. This slide gives an overview of different consortium of microorganisms and how they contribute significantly to organic P mineralization and inorganic P solubilization. Now, as I said earlier on, all these processes are enzyme catalyzed processes, and microorganisms, as they secrete these enzymes help to transform all these different pools of inorganic phosphate within the soil. Now, the activities of all these enzymes are controlled by genes. So for the focus of this research, we aim to quantify the expression of 4D genes, which is secreted by alkaline phosphatases, and also inorganic pyrophosphatases, which is controlled by the PPA gene. Now, another key aspect of my research is the use of dredge materials as agricultural amendments. What are dredge materials? What are these sediments? What are they made of? During agricultural runoffs, nutrient-rich top part of the soil are being washed into nearby water bodies, and this causes a buildup of sediment over time. As these sediments build up, they make it very difficult to maintain navigation channels to ports and harbors. Agencies such as the US Army Corps play a significant role in dredging to maintain navigation channels to ports and harbors. Annually in northern Ohio, 1.5 million tons of sediment are being dredged. All these dredged materials were previously openly disposed into Lake Erie, which led to the development of harmful algal blooms. The view here you see here, or the map you see here, is the satellite view of the Western Lake Erie Basin. The Western Lake Erie Basin. You can see how these blooms are being spread widely. Now, this led to an incident in August 2014 where Ohio Toledo went into a state of emergency because of the infiltration of harmful algal blooms in their water system. Because of this, open water disposal has been prohibited by law since July 2020, and this has led to the establishment of a facility known as the Facility 3 to store all these dredged materials. Just imagine if 1.5 million tons of sediment are being dredged annually in northern Ohio, there is the need to look out for beneficial uses of all these dredged materials. One of the beneficial uses with the focus of my research is to use all these dredged materials as agricultural amendments. Now, previous research has shown that dredged materials kind of has higher soil parameters that are very essential in determining the soil health. Parameters such as cation exchange capacities, which highlight the fact that the soil has the tendency to retain nutrients within the soil, and also dredged materials has shown to have higher bioavailable calcium concentration. It has also shown to have higher total potassium concentration within the soil. This brings us to the main research question, my research question. How does the amendment of dredged materials influence phosphate recycling in soil? One of the hypotheses is that applying dredged materials may alter soil microbial activities and influence the overall soil health. Now, in an attempt to answer all these questions, my research is a greenhouse-based experiment which integrates different approaches such as solid characterization, isotopic analysis, and genomic analysis. Now, with the solid characterization, we have outsourced samples for total nitrogen and total phosphorus, and also we have outsourced for basic soil tests such as pH, bioavailable P, bioavailable calcium, and cation exchange capacities. Now, in-housely, we have characterized for total organic carbon and heavy metals, as well as cations. With the isotopic analysis, we aim to measure the rate of recycling of phosphate using oxygen isotopes. With the genomic analysis, we have extracted DNA and RNA, which will be further subjected to 16S rDNA amplicon sequencing to be able to determine the different types of microbial consortium involved in phosphate recycling within the soil. And with the RNA, we aim to subject them to RT-qPCR, that is reverse transcriptase quantitative polymerase chain reaction, to be able to measure and quantify the different genes with respect to phosphate recycling in the soil. The source of soil for this research was from the Ohio Agricultural Research Development Center, Northwest Agricultural Research Station, located in Castor, Ohio. The field is made up of Hottville silt clay loam soil with slope ranging from 0 to 1%. With the focus of this research, this field has 3 main treatments. However, with the focus of this research, we collected soil samples from Section A and Section B. Section A has dredged materials, whereas Section B has no dredged materials. All these soil samples were collected and transported to the greenhouse of the Bowling Green State University, and they were homogenized, and then foreign particles were removed, and they were then redistributed into the mesocosms. They were subjected to watering, that is, to awaken the microbial communities within the soil. We planted soybean within all these mesocosms to stimulate the microbial communities within the soil, and as you can see from this picture, the soybean did very well in terms of growth. Moving to my preliminary results, this slide shows my preliminary results for total organic carbon and total nitrogen concentration within the 2 different treatments. Now you can see I have time 0 and 120 days after harvest. With the time 0, it explains the fact that after the soil samples were collected from the field, immediately we characterized them for total organic carbon concentrations, and after the greenhouse experiment, that is 120 days after harvest, we characterize them for total organic carbon concentration again to see the balance. Now, with respect to dredged materials, you can see that averagely we have considerably higher total organic carbon concentration in dredged materials compared to no dredged materials at time 0. At the end of the greenhouse experiment, we still had considerably higher total organic carbon concentration within these treatments. With total nitrogen concentration, we can see that averagely at time zero, we had considerably higher higher total nitrogen concentration within the dredge materials compared to those without dredge materials. However, at the end of the greenhouse experiment, we realized that there was a sharp decline in total nitrogen concentration in the dredge materials. Now, there could be two explanations that can be given for the sharp decline of total nitrogen concentration at time zero and 120 days after the harvest. One of the reasons could be that the dredge materials had more microorganisms involved in denitrification process. That is why we can observe a sharp decline in total nitrogen concentration. Another reason could be that there was more incorporation of nitrogen into the biomass of the plant, even though we have not done that quantification yet. These are some of the speculations we can make based on the trends we are seeing. Just to make this clear, this data has not been statistically analyzed yet. This is just a preliminary data. Moving forward, from the mesocosms, that is the treatment with dredge materials and those without dredge materials, we were able to recover some roots and subject them to winerizo analysis. With winerizo analysis, it gives us an idea of the root morphology and how well the roots have grown in terms of tips and then in terms of length as well. Now, as we can see here, dredge materials tend to have finer roots compared to those without dredge materials. Previous research by Brigham in 2021 showed that with the increment in with the increase in dredge materials, there is more robust and fine roots compared to those without dredge material. So as you can see here, with 100% dredge materials, we have more robust and finer roots. With 20% dredge materials, it decreases. However, it increases with the increase in dredge materials. Now, in terms of the average root length, we can see higher root length in terms of dredge materials compared to those without dredge materials. Moving forward, with cation exchange capacity and bioavailable calcium considerably at time 0 and 120 days after harvest, we had higher levels of cation exchange capacity and higher levels of bioavailable calcium, as research has shown that literature has shown that the driving force of cation exchange capacity is calcium concentration. So it is very evident here that with the higher cation exchange capacity, we still have a higher trend of bioavailable calcium in dredge materials compared to no dredge materials at time 0 and 120 days after harvest. Moving forward, this slide shows a preliminary results of the total phosphorus concentrations and the cation exchange capacity, just to make references. Now, with total phosphorus concentration at time 0, considerably, we had higher total P concentration in the mesocosms without dredge materials compared to those with dredge materials. However, at the end of the greenhouse experiment, we observed a sharp decline in the total P concentration in the mesocosms without dredge materials. Now, the reason could be because there is low cation exchange capacity in the treatment without dredge materials. Now, with low cation exchange capacity, the soil has less tendency of being able to retain nutrients within the soil. Hence, all these, the speculation here we are making is that the total phosphorus or higher portions of the total P were being leached out from the bucket. That is why we can observe sharp decline in total P within the mesocosms without dredge materials. Now, the next step for this research is to continue with the isotopic analysis to be able to measure the rate of recycling. Now, just to give a snapshot of the isotopic measurement here we are trying to do, we aim to shift the water isotopic equilibrium from 11 per mil to 33 per mil. Now, the key takeaway point here is that the shorter the time taken to reach the new shifted isotopic equilibrium, the faster the rate of recycling. So we are hypothesizing that based on all these beneficial parameters that dredge materials have, we anticipate that dredge materials will take a short time to reach the new shifted isotopic equilibrium. Hence, there will be a faster rate of recycling in the mesocosms with dredge materials compared to no dredge materials. Now, we also, with the genomic aspect, we aim to quantify the expression of specific genes such as FOD, that is alkaline phosphatases. We also aim to quantify the expression of APPA, that is inorganic pyrophosphatases, using reverse transcriptase quantitative PCR. I would like to end my presentation by giving a brief agricultural implication of this research. The addition of dredge materials in organic farm soils have shown increase in parameters such as cation exchange capacity, total organic carbon concentration, and calcium concentrations. The use of nutrient-rich dredge materials might improve nutrient cycling and enhancing the overall soil health, which will then lead to sustainable agricultural practices. I would like to acknowledge the expertise of my professors, Dr. Kevin McCloney and Dr. Angelica Vasquez Ortega, and not to forget Dr. Chris Ward for their wonderful contribution towards this research. I would like to also say a big thank you to the BGSU research team, and also acknowledge all the funding agencies that has been very supportive throughout this research. These are some of the few references I used, and thank you.